everybody from VCU. I can see that Jinshao and Sheena are from Kentucky, and then you and caller 1 and caller 2. I hope Imangi has joined from New York. Okay, so we have Professor Nancy Dahms from Department of Biochemistry at College of Wisconsin. She's going to, she's an expert on lysosomal storage disorder. And she agreed to give the lecture. I used to give it up until now, but she has much more broader expertise, and I thought it would be really good to have her. She's also part of our K-12 grant application. So we have her, and then we have Nancy Hoffmeister, who is from Blood Research Institute, going to follow up after about an hour or so. So Nancy is, if you can wave your hand, Nancy, there she is, okay, and she has, all right, very good. Thanks, Nancy.
Okay, you're welcome. So everyone can hear me?
Yeah.
Okay, great. Okay, so today, as Dinesh said, we'll be talking about lysosomal storage diseases, and you've heard throughout the course a lot about different glycans and a bit about their synthesis, and we're going to focus on their degradation. So I've listed the learning objectives here for today's session. We'll learn a little bit about the degradation of glycoproteins, glycolipids, and glycosaminoglycans. We'll see that this occurs in the lysosome, and we'll look at these acid hydrolases that carry out this catabolism. And then we'll go into some of the general features and symptoms of patients with lysosomal storage diseases, and then give you both current and future directions about how these lysosomal storage diseases are treated. So going into a little more detail about the outline, again, we'll be going through the catabolism or degradation of glycoproteins, glycolipids, and glycosaminoglycans. And we'll see that this occurs in the lysosome. So I'll give you some background about the lysosome, what its function is, how it's generated. And so we'll talk about how these hydrolytic enzymes, these acid hydrolases, actually get delivered to the lysosome, and we'll see that they acquire a unique tag, a carbohydrate tag, and they're recognized by lectins, and you've already been introduced to lectins in the course. And we'll also see that if this process is disrupted, it results in a disease called mucolipidosis 2. Then we'll transition overall into looking at when you have defects in the degradation or catabolism of these glycoconjugates, that there's a family of diseases called lysosomal storage diseases. And we'll go over how they're classified, some of the major clinical symptoms, how they're treated, and I'll give you a few specific examples of lysosomal storage diseases that we'll go through. Okay, so that kind of sets the stage for this session. So let's first look at the lysosome. So what's the lysosome? So I like to refer to it as the recycling center of the cell. So lysosomes, they are membrane-bound organelles. They're found in virtually all cell types. They contain a repertoire of about 60 different acid hydrolases, such as proteases, glycosidases, lipases, that are able to degrade glycoproteins, glycolipids, and glycosaminoglycans. Why, again, I call it the recycling center of the cell. So, for example, in the lysosome, if you degrade a protein, they'll degrade it into amino acids, and then there are transporters in the membrane of the lysosome to allow those amino acids to leave the lysosome, and then they can be reutilized for new biosynthetic events. Same is true for monosaccharides. If you're degrading a glycoconjugate into individual monosaccharides, those can leave the lysosome and then be reutilized. In the case of, say, glycogen to glucose, that can enter, say, into the TCA cycle for energy production. So again, this is a very critically important organelle. These hydrolytic enzymes, they're often referred to as acid hydrolases. These are soluble proteins, and why they're called acid hydrolases is because these enzymes work optimally at the acidic pH that's found in the lysosome. And why these compartments are acidified is it because they have associated with them a type of ATPase called a Z-type ATPase, in which upon ATP hydrolysis, protons are pumped into the lumen of this organelle to acidify it. So that's kind of the big picture here of the lysosome, of what it is. And it's composed of these 60 or so different soluble enzymes that carry out its degradative properties. Now, looking at the function of the lysosomes, there are many important functions, and I've just summarized a few of them here. I just wanted to highlight that back in the 1950s, Glycan Degradation discovered the lysosome, and then he was acknowledged for that discovery in 1974 with a Nobel Prize. So some of these critical functions that the lysosome carries out, I've listed here. For example, they dispose of abnormal or aggregated proteins as well as defective organelles, such as mitochondria, for example. They can downregulate signaling pathways, such as EGF receptor. They can release endocytosed nutrients, such as cholesterol from lipoprotein particles. They're involved in destroying or inactivating pathogenic organisms by phagocytosis. And for example, macrophages are one of the key cells in the body that carries out phagocytosis to destroy various pathogenic organisms. And also they're important for cellular survival by a process called autophagy. Now, I've listed here there are three major ways that molecules can get to the lysosome. So by, as I mentioned, phagocytosis, where there's an engulfment of, say, a bacteria into the cell, by a process called endocytosis, and then autophagy. So autophagy, what it means is self-eating. Again, it's a major catabolic process that is really a housekeeping process that occurs all the time. So the cell has mechanisms to sense when an organelle is defective, and so it needs to be gotten rid of from the cell, as well as aggregated proteins. Again, there's mechanisms that the cell can do that. And again, it allows the cell to then turn over those molecules for new biosynthetic events. Now, autophagy is a process that is tightly regulated. And if a cell is starving, this process can be upregulated to allow the cell to survive by self-eating a little bit until the environment changes. And I also wanted to mention that Yoshinori Oshomi received the Nobel Prize for his work on studying this autophagy process. So as you can see, the lysosome is really critical for many, many important processes in the cell and in the organism. So the question is how do we make a functional lysosome? As I mentioned, the lysosome contains about 60 different soluble enzymes. So how do we then package those lysosomes, those enzymes into the lysosome? So all proteins start their synthesis on cytosolic ribosomes. So again, the question is, how do you target or deliver these 60 or so lysosomal enzymes from their site of synthesis to the lysosomes, because that's where they function. And so the big picture that I've outlined here is that these lysosomal enzymes, these 60 or so different enzymes, are uniquely tagged or have this zip code of mannose-6-phosphate. So this is a unique carbohydrate mark that's placed on these enzymes. And they're recognized by receptors or these P-type lectins, and there's two of them, okay? So one's smaller, one's larger. And so again, we've got 60 or so different types of lysosomal enzymes. They all have this mannose-6-phosphate tag on them that allows them to be recognized by the P-type lectins. And why they're called the P-type lectins is because they recognize phosphorylated glycans. So that's how they got their name, okay? So again, the big picture is that for this specific type of trafficking, you have specific lectins that will recognize this unique tag, this mannose-6-phosphate tag. So I thought I'd bring this up as an analogy that is useful to think about this process that we'll be talking about in a little more detail. So this analogy is write, read, and erase. And so when you think about writers, we think here in the case of glycans, glycosyltransferases. So these are the enzymes that make that specific glycan. Okay, so those are the glycosyltransferases. They're adding that specific mark. The readers are the ones that are interpreting the mark. And you've already seen in the course that there's lots of different types of glycan structures. And the question, sometimes the challenge, is to understand how the cell can interpret all of that different information that's encoded into a glycan structure. And so we have these glycan binding proteins. You've seen this diagram earlier that Dr. Desai had shown you about these glycans shown here, and then we have these glycan binding proteins, these readers, or these lectins, okay? So that's what's going to interpret the mark. And then we also have erasers, those that remove the mark. In this case, these are glycosidases, okay? So a nice analogy to think about what we're talking about, and I'll come back to this terminology as we go through this targeting process. Okay, so how do we, again, mark these lysosomal enzymes, these acid hydrolases, with this unique mannose-6-phosphate tag? So in the first step, we have a glycosyltransferase. And so what I've identified here is that all of these lysosomal enzymes, they are glycoproteins that have end-linked glycans, okay? So here in purple is depicting the protein portion of the molecule. This is a high mannose end-linked glycan. And we have this unique glycosyltransferase termed N-acetylglucosamine 1-phosphotransferase that utilizes the sugar nucleotide, UDP-GLUCNAC. And this particular glycosyltransferase is able to recognize the 60 or so different lysosomal enzymes that are going through the Golgi, and then add actually an N-acetylglucosamine and a phosphate onto a specific mannose to form what's termed as phosphodiester. So this is key. This enzyme, this glycosyltransferase, again, is able to distinguish the 60 or so different acid hydrolases from basically thousands of proteins that are going through the Golgi and mark it with this unique tag. So the second step, there's a glycosidase that removes the N-acetylglucosamine, and you're left with this mannose-6-phosphate recognition marker, this zip code, this tag, however you want to call it. So this is that unique tag that's placed on a lysosomal enzyme. This tag is then recognized by a receptor. So these are these P-type lectins. And I mentioned there are two of them. They're called mannose-6-phosphate receptors. One is a smaller one, and then one is a larger one. And again, they're able, these proteins, these are integral membrane proteins, are able to recognize that mannose-6-phosphate tag that's found only on these lysosomal enzymes. So again, this process, this tagging process, occurs in the Golgi, first by the glycosyltransferase and then by a glycosidase to have this mannose-6-phosphate recognition marker. We then have these receptor molecules, these P-type lectins, that recognize only those proteins that have that tag. So again, there are thousands of different proteins that are going through the secretory pathway and through the Golgi. And again, these P-type lectins, that's what they're recognizing, only those molecules that have mannose-6-phosphate on them. What the receptors do is then bind those molecules, those lysosomal enzymes, and they carry them to an endosomal compartment. In the endosomal compartment has a more acidic pH that allows the receptors to release the lysosomal enzymes, and they're packaged into the lysosome where they function. The receptors then recycle back to the Golgi and repeat this process. So they can carry out this process hundreds and hundreds of times during their lifespan. So these receptors have a half-life of about a day. So again, they can carry out this pathway hundreds and hundreds of times to take newly synthesized lysosomal enzymes from the Golgi and deliver them to these endosomes and lysosomes where those enzymes will function. Okay? So that's kind of a big picture of how this trafficking process occurs. Now, we know actually a lot about these P-type lectins. So my laboratory has carried out a number of structural biology types of studies on these two proteins using X-ray crystallography and NMR spectroscopy. And again, there's a lot of details here that I obviously won't have time to go through, but the big picture is we know how the proteins are interacting with very high affinity with mannose-6-phosphate. So here we have a close-up view of one of these P-type lectins. We have mannose-6-phosphate. We have the mannose ring here. The phosphate group is shown here. And we know by these high-resolution structures, we know all the different amino acid residues that are interacting and forming hydrogen bonds with this mannose-6-phosphate. And what's a little different from some of the other lectins you may hear about, the P-type lectins are unusual in that they're able to bind this glycan with high affinity, okay? And again, part of that because we understand the types of interactions that are occurring in this binding pocket. But that's all the details I expect you to get out of this particular slide. Again, we have two of these different receptors. They bind very tightly and very specifically to a phosphorylated sugar, mannose-6-phosphate. Now, I've given you information about how important the lysosome is, and I wanted to give you some information of how we know that this targeting process is also a critical process to the health of individuals. And so we have a system where we have a writer, a glycosyltransferase that makes this mannose-6-phosphate tag. We have P-type lectins that can read and deliver these enzymes to the lysosome. But is this process really important? Well, we know if we disrupt this process, it results in patients that have a disease called mucolipidosis 2, which is a lysosomal storage disease. Now, what happens with patients with this disease is that the defect is in the enzyme that puts on the tag. So it's in the defect in the glycosyltransferase, the writer, that makes the mannose-6-phosphate tag. So patients that are deficient in this enzyme, none of their 60 or so different lysosomal enzymes will acquire that mannose-6-phosphate tag. Since they're not marked for delivery to the lysosome, since these are soluble enzymes, they're secreted from the cell. So these patients basically have lysosomes that are pretty much devoid of any acid hydrolases. And as you can imagine, then they have massive storage or accumulation of lots of different macromolecules because they don't have the degradative enzymes to degrade those molecules. And so you have this accumulation, the storage, why it got a name of lysosomal storage disease, in their cells. And these patients are quite, as you can imagine, quite severely affected given all the important functions that a lysosome carries out that I showed you earlier. And so these patients will have skeletal abnormalities, cardiorespiratory complications, and typically these children die by the age of 10 years. And so again, this is very critical targeting pathway that you have to have a functional lysosome in order for a person to survive. And this actually was the first shown link between glycoprotein biosynthesis and human disease. Okay, so I thought I'd stop here for a minute and see if anyone has any questions about this targeting pathway.
I have a question.
Okay, go ahead.
So does this mean that these patients will not have any acid hydrolases or won't have specificity of acid hydrolases?
So the question is about their lysosomes, whether or not they do or don't have any acid hydrolases. So again, their lysosomes are severely depleted with acid hydrolases. There are a few cell types that have some acid hydrolases, and up to this point, we still don't understand the pathway, how a few cell types can have some acid hydrolases. That's going into some additional details. But again, most of their cells, most of their lysosomes are quite devoid of acid hydrolases. So they're not able to carry out normal degradative metabolism.
Thank you.
Does that answer your question?
Yes, it does. Thank you.
Okay. Any other questions?
For without the phosphate, is the binding completely abolished, or is it still partially there?
Okay, so that's a good question. So the question was, if these patients don't have this phosphate, is it really completely abolished? And so we've done a lot of biochemical studies to measure the binding affinity. And so the phosphate part is absolutely critical for high affinity binding. So you go from, if you have the phosphate there, the binding affinity is in the nanomolar range. Without the phosphate, you go to the micromolar range. And so that is, over 1,000-fold drop in affinity, and that really limits the ability of these T-type lectins to deliver and bind tightly enough to carry these molecules from the Golgi to different compartments. So yes, so in all of our structural studies, that phosphate is absolutely critical. It's a good question.
Glycan. I actually have one more question real quick. So, so it looks like when these mannose-6 phosphate receptors actually bind the mannose-6 phosphate, they essentially a bud is formed, and that forms the endosome. Is that what I'm seeing?
Correct. So when molecules go through the secretory pathway, there are different stacks of the Golgi, and you have membrane-bound vesicles that bud off the Golgi, and then they will fuse to the endosome. Vesicles will bud off the endosome and fuse to the lysosome. Yeah, so you have those processes going on.
So is this how the V-type ATPases actually get to the lysosome, and then they can create those acidic conditions?
So that's a good question. That I don't have a good answer for how the V-type ATPases are delivered. It would go through these membrane-bound vesicles, but to my knowledge, they do not have mannose-6 phosphate. If that's what you're asking, it's really the soluble enzymes that have the mannose-6 phosphate on them. And V-type ATPases, they're membrane-bound molecules.
Okay, thank you.
Sure. So presumably they have a different type of targeting pathway. Okay. Any other questions? No. Okay. Those are all great questions. Okay. So I mentioned here that this mucolipidosis 2. This is a defect in this glycosyl transferase that causes a lysosomal storage disease. So I now want to talk more about lysosomal storage diseases. So this is an example of one of them. But in general, what are lysosomal storage diseases? They're a family of progressive and degenerative disorders that affect catabolism of glycoproteins, glycolipids, and glycosaminoglycans. There's actually about 70 or so lysosomal storage diseases, and about 50 of them or so are affecting a particular hydrolytic enzyme. So the previous one I showed you, mucolipidosis 2, this is affecting the tag that's put on the enzyme. Most of the other types of lysosomal storage diseases, they're defective in a single hydrolytic enzyme. I mentioned there's about 60 or so of them in the lysosome, okay? And many of them are glycosidases. And we'll go through a couple different examples. I'll give you an example of Fabry and Pompe. So again, what happens in lysosomal storage diseases, it's affecting the catabolism of a particular molecule if you're defective of a particular hydrolytic enzyme. And this results in intracellular accumulation of undegraded material. And again, some of the types of symptoms that patients have, they can have skeletal deformities, mental retardation, heart disease, decreased life expectancies. These are common features among many of these lysosomal storage diseases, and they affect many different organs. Now, what I've plotted here is the disease phenotype, so the onset of the disease, and this is the amount of enzyme activity that's left. So again, what's happening in these patients is they'll have a mutation in, say, a particular hydrolytic enzyme. And that mutation could leave that that protein, that enzyme, with virtually no enzyme activity. It could have a couple percent enzyme activity, 20%, 30%, 50%. And what I've shown here is that the disease severity and the age of onset is inversely proportional to how much residual enzyme activity you have based on the mutation in the gene. So if you have a mutation that leaves you with virtually no activity, you can envision, and this is what happens, is that the onset of the disease occurs at birth or very soon after, and is quite severe. Other mutations, you might have more residual enzyme activity, so you might not see the onset of symptoms until childhood, teenage years, or if again you even have a little bit more, it might not be till adulthood. And there's a certain threshold, depending on the enzyme, that an individual could be asymptomatic. So again, these are quite diverse types of phenotypes that are seen in these patients. These diseases are inherited most often by an autosomal recessive type of phenotype. That means both alleles have to have the mutation, and there are a few that are X-linked. So again, mutation is just on the X chromosome, and males are much more severely affected. Again, there are many different types of lysosomal storage diseases. Again, there are about 50 of them that affect hydrolytic enzymes. Overall, their incidence is about 1 in 6,000 live births. So as a group, they're among the most common genetic disorders in childhood. Okay? And as I mentioned, the presentation is quite heterogeneous depending on how much enzyme, residual enzyme activity you have based on that mutation. Okay. So we have lots of different types of lysosomal storage diseases. They have different names. And how they're classified is based on the substrate that they accumulate. So again, we have for mutations that affect the degradation of glycosaminoglycans, they're grouped together and they're called mucopolysaccharidoses. Okay? Some examples are Hunter and Hurlers. If a patient cannot degrade glycosingolipids, they're classified as singolipidoses. And we'll go through the example of Fabry disease. If you cannot degrade lipids, they're termed lipidoses. If you cannot degrade glycoproteins, so the N-linked glycans, for example, they have a different terminology, glycoproteinoses. And then we just looked at mucolipidosis 2. This is a defect in enzyme localization. So this is where, again, that particular glycosyl transferase is where the mutation is, rather than these hydrolytic enzymes. And this is, again, a disordered enzyme location. Okay? And I've given you a website that you can find, if you're interested, all the different names of all the different lysosomal storage diseases. So if we look specifically, say, at the singolipidoses, so what do we mean when how are we degrading glycolipids, glycosingolipids? So here I have an example of a particular glycosingolipid. This happens to be a ganglioside, GM1. We have the ceramide backbone. We have these different sugars indicated here. Again, we have sialic acid, negatively charged. And what we see when this glycosingolipid is degraded, it occurs sequentially by the action of exoglycosidases that remove the sugar one at a time from the non-reducing end of the glycan. So we're starting out with this molecule. We have a particular glycosidase here, beta-galactosidase, that removes this yellow sphere. This monosaccharide here is removed. Then the next exoglycosidase can act to remove an N-acetylglucose, the lactosamine. We have a neuraminidase that can remove the sialic acid, another galactose, the glucose, and then we're left with the ceramide. Okay? So this is a sequential process that occurs by the action of exoglycosidases. And so you can envision, if you have a deficiency of one of these enzymes, the substrate upstream of that enzyme will accumulate. So, for example, we'll go through Fabry disease. If we have a deficiency of that enzyme, this particular glycosingolipid called GB3 will accumulate because we're missing that enzyme. And again, it's a sequential process for how we degrade it. Now, a similar process occurs if this were a glycoprotein. Again, in the lysosome, we would have proteases that would degrade the protein part of the molecule into amino acids. And again, for the end-linked glycans, we'd have the same type of process where we have the sequential action of exoglycosidases that would remove the sugar one at a time. Okay. Now, when we think about diseases, we also consider, when we look at the symptoms of patients that have lysosomal storage diseases, it also depends, their symptoms will depend on the type and abundance of the molecule that is being degraded. So I've given you a couple of examples. So for those patients that they cannot degrade gangliosides, this is an example of a ganglioside here, a particular type of glycosingolipid, gangliosides are known to be highly expressed in neurons and in the brain. So those patients, if they can't degrade a ganglioside, predominantly they have neurological disorders because that's where so much of this material is building up. And eventually it will cause dysfunctional lysosome and cellular death. In contrast, if glycosaminoglycans are highly abundant in the MPS diseases that I'll tell you a little bit about, predominantly these individuals have musculoskeletal disorders. So again, in part, it depends on what material is being accumulated and the type of tissue where that particular substrate is highly abundant. So if we look, again, to go in a little more detail as an example for Fabry disease, again, Fabry disease is an example of a single lipidoses, and it's deficient a particular enzyme, alpha-galactosidase A. So this is the molecule, this glycosingolipid, that's going to accumulate in those patients. So Fabry disease is one of the few examples of a lysosomal storage disease that's X-linked. Originally, it was thought that this particular lysosomal storage disease was relatively rare, about 1 in 100,000. However, in the last few years, a few of the states in the United States, as well as a few countries, have mandated newborn screening. And it's been shown that Fabry disease is now the most common of the lysosomal storage diseases, and actually with a relatively high incidence rate. So in the state of Washington, when they've done these newborn screenings, 1 in about 6,000 newborns have Fabry, have mutations in alpha-galactosidase A. Illinois, about 1 in 8 or 9,000. And again, this is showing the data from Illinois newborn screening to show that Fabry disease is the most common. Again, not surprising because, as I'll show you in a minute, patients that have Fabry disease, they will live into their reproductive years. And so in comparison to some other lysosomal storage diseases where the death can be quite young, these patients are living and can reproduce. So it's not surprising that they may be more abundant than maybe some of the other lysosomal storage diseases where death occurs quite young. Now again, just to reiterate that many of these lysosomal storage diseases can affect many different types of organs. For the example of Fabry disease, where again we have a glycosingolipid that's accumulating, we see that we have certain symptoms appearing early in childhood, such as pain. They have cloudiness of the cornea and the lens. As we go to adolescence, some other symptoms now appear. They have pain in the GI, gastrointestinal distress. They have certain angiokeratomas. These are lesions in the skin. But then as we go into adulthood, and these are the symptoms that shorten their lifespan by about 10 to 20 years, they'll have strokes, they have renal failure, and heart attacks. Okay? And so again, this is again a good example of how these diseases are progressive because with age, their cells are accumulating more and more of that particular glycosingolipid. So again, the enzyme that's deficient is called alpha-galactosidase A. This is an example of the crystal structure that we know about that particular enzyme. And again, if you're missing that enzyme, this particular glycosingolipid called GB3 is accumulating in their tissues and in their serum. Now, in Fabry disease, over 700 different mutations have been identified in that gene. And again, Some of these mutations cause complete absence of any enzyme activity. Some have quite a bit of enzyme activity, but that enzyme could be unstable or it might not get to the right place. Okay? So we have a wide range of what can cause deficiencies in that activity. So again, if you don't have any enzyme activity, it will have massive accumulation of glycosphingolipids, and the onset will happen earlier for those individuals, rather than if you have quite a bit of enzyme activity, you might not see symptoms until later on in adulthood. Now, how do we treat these lysosomal storage diseases? Two of the earliest treatments for patients with lysosomal storage diseases are enzyme replacement therapy and hematopoietic stem cell transplantation. So actually, enzyme replacement therapy was first carried out in clinical trials back in 1973, but it wasn't until 1991 that the first FDA-approved enzyme replacement therapy became available to patients. And I'll go into that in a little bit, but what's happening here is patients are given the recombinant enzyme that they're deficient, and this occurs by intravenous infusion, typically every 2 weeks. Okay, so I've listed some of the limitations of this type of treatment. Again, it's not a cure. At best, it slows down symptom development, but there, again, can be some issues. Again, you're giving a protein to the patient. Generally, large proteins don't cross the blood-brain barrier, so if you have certain lysosomal storage diseases that affect the brain, this is not a very effective treatment. Hematopoietic stem cell transplantation has its own risks. It's been used for a few of the different lysosomal storage diseases, and clinical trials were first done back in 1981 for this type of treatment. Now, looking at enzyme replacement therapy, why I talked to you about the P-type lectins is that for all of the lysosomal storage diseases, there are actually about a total of 70 of them. There is only FDA-approved treatments for a total of 11 of those lysosomal storage diseases. And of them, enzyme replacement therapy is by far the mainstay of treatment, but it treats only 9 of them. Okay, so we have a long way to go to treat these patients. And for the vast majority of these enzyme replacement therapies, how the enzyme gets into the cells is by the P-type lectins. So these enzymes that they're made recombinantly, they have to have mannose-6-phosphate on them to be taken up by the endogenous P-type lectin. Okay? And so again, we have intravenous infusion every 2 weeks with this recombinant protein that's then taken up by the P-type lectin, and then it gets delivered to the right place. As you can imagine, this is a very expensive therapy to give a protein that often. So again, prices per year can go anywhere from $250,000 to almost $500,000. So it's quite expensive treatment.
Are there any complications? Like, you think about this, they just have a lot of phosphate receptors. They don't have any kind of immunologic.
Right. So the question is, what are some of the side effects or issues? So I mentioned a little bit for those patients that do not make any active enzyme. Again, if you think about it, this is like immunizing like a rabbit to make antibodies. You're giving a protein that they've not seen before. So oftentimes those patients can develop neutralizing antibodies because they're recognizing that protein as foreign. There's other infusion types of issues, but I don't know specifically about the macrophage issue.
The body is foreign. Considering it's not usually floating around like that.
Correct. But again, so for some of the patients, up to 50% of them can make neutralizing antibodies because they're recognizing that protein as foreign. Okay. So again, it's not a perfect treatment, right? But that's been the mainstay. Now, as you can envision, as I mentioned, there are so many lysosomal storage diseases that don't have any FDA-approved treatments, and there are a number of preclinical and clinical trials that are underway for a number of these lysosomal storage diseases. So we have, and I've given you examples here that are currently ongoing. So we have ways to try to improve the efficacy of enzyme replacement therapy. So how do we make it better? Do we put more mannose phosphate on it? Do we do something different to make it better? Do we get it to go across the blood-brain barrier? Very recently for Fabry disease, a pharmacological chaperone therapy has now gained FDA approval. So this is an example where the patients do make a mutant enzyme, but it has low enzyme activity. So this small molecule, which is an amino sugar, binds to the active site, stabilizes that enzyme, and helps it get to the lysosome, and then this molecule releases. So that's why it's called a pharmacological chaperone. Again, to handle those many, many different mutations. As I mentioned, Fabry disease, over 700 different mutations have been identified. Here's an example where the patients are making an enzyme. It's just not quite that active. This is a way to help to stabilize that enzyme, get it to the lysosome. There's other things that's called substrate reduction therapy. As I mentioned, if certain cell types make a lot of particular substrate, if you can reduce the biosynthesis, actually you can help mitigate some of those symptoms. Of course, there's clinical trials and preclinical trials for gene therapy, mRNA therapy. Actually, back in 2017, gene editing using actually a zinc finger nuclease gene therapy was used on patients with MPS 1 and 2. So we have lots of things that are ongoing to try to treat so many patients that really don't have anything available to them, and those that we're trying to get to the stage that we could cure the disease by gene therapy. And I've given you some information here, actually from Dr. Desai, who provided this information. There's lots of interest in the biopharma industry, again, because these typically are very crippling diseases. We've got 5-year exclusivity guaranteed by the FDA. Again, it's lucrative for many of the pharmaceutical companies to try to generate these types of molecules. There's lots and lots of active research going on in clinical trials to try to improve the lives of these patients. So I've just given you a list of the current FDA-approved treatments. Again, the mainstay is enzyme replacement therapy. That's treating 9 of these different lysosomal storage diseases. We also have substrate reduction therapy. This is an oral treatment. This is intravenous infusion. We have a chaperone therapy, oral, and then another oral for storage reduction therapy. So again, I don't expect you to know the names of these different compounds, but just know these are the different FDA-approved treatments that are available to patients, and we have lots more that are in the works in clinical trials. So finally, I just want to mention how glycosaminoglycans are degraded, and you'll hear much more from Dr. Desai about the biosynthesis of glycosaminoglycans. But their degradation is a little more complicated in that glycosaminoglycans, as you know, have negative charge, and they're heavily sulfated. And so, in contrast to what I showed you with glycolipids, where the sugar is removed, before you can remove one of the sugars from a glycosaminoglycan, you have to remove those negative charge. So we have additional enzymes called various sulfatases that remove the sulfate molecule, and then we can have another enzyme that removes that sugar. So again, we're removing this sulfate molecule. Now we can use an iduronidase enzyme to remove this iduronic acid. Now another sulfatase has to come in for that next sugar to remove those modifications, another modification. Then we can, again, remove the sugar, again, remove the modification, and so on. So that's why it's a little more complicated, if you will, because there's these additional decorations, these negative charges that need to be removed before we can remove the sugar as we're degrading the glycosaminoglycan chain. Now again, for glycosaminoglycan degradation, the family of lysosomal storage diseases is called the mucopolysaccharidoses. These are some of their major clinical manifestations, and musculoskeletal deficiencies and abnormalities are quite common among all the different MPS diseases. They also have coarse facial features, thickened skin. They can have developmental effects, and also cardiomyopathy, coronary artery disease, for example. And this is a table, again, of the different MPSs, the mucopolysaccharidoses. We've got, again, different enzymes that remove sugars, as well as some of the enzymes that affect the negative charge, the sulfate, that can be defective. But again, a major feature of most of these mucopolysaccharidoses is musculoskeletal defects. And then finally, I'd just like to leave you a little bit about Pompe's disease and to let you know that not only is pharmaceutical companies and many researchers interested, but Hollywood also loves glycans. So back in about 2010, a movie came out called Extraordinary Measures. And this was based on a true story of a man here played by Brendan Fraser, in that he had 2 children that were diagnosed with Pompe disease. Pompe disease is a lysosomal storage disease in which glycogen accumulates. So these patients are defective in a particular glucosidase and they can't degrade glycogen. The defect in these patients is quite severe because it affects muscle. So it affects the heart and the diaphragm. Oftentimes these children have to go on ventilators when they're in their early teens, or if not early. And death usually comes quite early without enzyme replacement. So this was about the time in this movie was before there was any FDA approval. And so this is a great bench-to-bedside story of how the enzyme replacement therapy was being developed for Pompe's disease. And I'm a big Harrison Ford fan, so this was great for me to see them on the whiteboard showing a glycan structure, talking about the P-type lectins and enzyme replacement therapy. So something a little fun if you're interested. You can probably still find this movie somewhere and look that up. So it gives you a little bit more about, you know, all the work and effort that took to get to the stage of having FDA-approved therapy. And then finally, I have a couple additional slides that I won't test you on, but if you're interested more about the a few of the lysosomal storage diseases that have defects in glycosaminoglycan degradation, these are provided by Dr. Desai, that talks about some of the diseases, a little bit more about the symptoms, and a little bit more about the FDA-approved enzymes and some of the issues with enzyme replacement therapy. But again, I won't be testing you on those details, but again, if you're interested, it shows you a little bit more about that information. Okay? So with that, I'll stop and see if you have any questions that I can answer for you.
Questions? Okay.
Nancy, I have a quick question. This is Joe Lau.
Hi, Joe.
Nancy, hi. So the enzyme replacement therapy, are there any different, what are the different strategies? You know, obviously you're just pumping the body full of a replacement recombinant enzyme floating around in the blood. What are the efforts to try to get these floating enzymes to be concentrated into the target rather than just freely floating that, and if that might improve the efficiency of the enzyme replacement therapy?
Right. So some of the strategies. So again, classically, what has been done, as I said, for most of them, is to make those enzymes in, say, Chinese hamster ovary cells that have mannose-6-phosphate, because again, this is a receptor-mediated process. By receptor-mediated cytosis, this normally allows that concentration effect to occur. So what efforts are made is to make a more highly phosphorylated molecule. So, for example, there is one deficiency for one of the MPS in which when that enzyme is actually made in, say, CHO cells, which is quite common in pharmaceutical, you use that particular cell line, only 3% of it acquires mannose-6-phosphate. So clearly, that's not very efficient for being taken up by the mannose-6-phosphate receptor. And so there have been efforts to chemically conjugate mannose-6-phosphate on enzymes. For Gaucher's disease, actually what's used is the mannose receptor. So that's the one exception to the general rule of using mannose-6-phosphate because of that cell type. They're targeting the mannose receptor for that particular disease in Gaucher's disease. What I didn't mention about this receptor, and let me go back, another strategy that's being used, let me just go back to my picture of the receptor. This receptor also binds a small polypeptide called insulin-like growth factor 2. As its name implies, it's like insulin, which is 67 amino acids. So there are a couple clinical trials ongoing in which they fuse this peptide onto the lysosomal enzyme. And again, this binds this molecule with very high affinity, but you're not dependent on mannose-6-phosphate. So another way, again, targeting this protein. Now, getting across the blood-brain barrier, some people just give a huge bolus of the protein to patients. Again, there's lots of issues with that. And others are looking at other types of receptor molecules to get across the blood-brain barrier. One of them is, I think, the LDL-related protein that people have been using to target to get across the blood-brain barrier. I don't know at this point how efficient or how good that is, but again, those are other studies of looking at more receptor-type molecules to do the concentrating for them for targeting to particular types of molecules. Does that help a little bit or get at your question, Joe?
That helps a lot. Thank you very much, Nancy.
Sure.
Do you know off the top of your head, roughly, when you talk about large doses of recombinant enzymes given to these people, what is the dosage? How much are these people being pumped with?
So say, for example, for Fabry disease, the one that's approved in the United States, it's a mg per kg, 1 milligram per kilogram body weight. The one in Europe is 0.2 milligrams per kilogram. Those are typical ranges. Some can be higher. So again, for example, if it's poorly phosphorylated, you have a significant fraction of that protein that's not going to be taken up very efficiently. One of the issues with Pompei's disease, so this is the disease where muscle cells are being affected. It's known that the P-type leptins are not highly expressed in muscle. So that's an issue, right? So you don't have a lot of the receptor there to begin with.
Right.
And so again, it's what people end up doing is trying to give even more higher doses to try to offset that. So whether or not there's other ways of trying to think, well, can we use maybe a different type of molecule to target muscle cells better? Is there a way to, say, upregulate the receptor that you're interested in? So there's all those things that people are thinking about and knowing what these limitations are. So not only do you have to worry about a tag in this case, mannose-6-phosphate, or using IGF-2, you also have to consider the expression level of the receptor that you want to target.
Thank you.
Sure, you're welcome. All great questions.
So Nancy, a quick question.
Yes.
What about sulfate recognition by the receptor? Mannose-6-sulfate.
Okay, so what we do know is when we've done inhibition studies, so for this receptor, there are 4 mannose-6-phosphate binding sites. They do differ. This one here is able to recognize mannose-6-sulfate. The other ones do not. It's not as high affinity. It's about a 20-fold poorer affinity than mannose-6-phosphate.
Wow. Interesting.
No, it is interesting, right? So this one, there's enough flexibility, if you will, in the binding pocket that it can accommodate. So typically just with looking at mannose-6-phosphate, typically the affinity might be 20 micromolar just for the sugar, whereas with mannose-6-sulfate, it's about 200 micromolar. So again, a 10 or 20-fold difference we see. But just with this binding site, not the other ones will not accommodate mannose-6-sulfate.
Thanks.